Well, thank you, Teresa, and good morning to everyone, and welcome to this virtual workshop. We wish we'd been able to see everyone in person, but we appreciate everyone who's registered and is attending right now. As Teresa said, my name is Michelle Campbell, and I lead stakeholder engagement and clinical outcomes in the Office of Neuroscience. And for the next few moments, I'm going to talk to you about how we envision how this Rare Disease Cures Accelerator will help inform, how that data will help inform rare disease drug development and what it can really do to help accelerate as we talk about the unmet needs we have in rare disease drug development. As Teresa stated, we have 7 million rare diseases, with only 10% of those rare diseases having an approved treatment. But we are making some strides in our rare disease drug development programs. Over the last few years in the Office of New Drugs, over half of all new molecular entities were in rare diseases. Two examples I wanted to share, being from the Office of Neuroscience, I wanted to show how we are doing rare disease drug approvals, including this past August, where we had the first oral drug approved to treat patients with spinal muscular atrophy. And this is the third drug that's recently been approved for SMA and the first oral. The second example I wanted to show you, that just this past summer, in both June and August, we had 2 different monoclonal antibodies that were approved to treat patients with neuromyelitis optica spectrum disorder. This drug, up until June 2019, had no prior approved therapies. And now we have 3 approved therapies for this rare disease. But with this growing trend and seeing more approvals happening in rare disease, we still have a lot of work to do. Last year, my colleague, Katie Donahue, presented a photo of a house upside down. And I wanted to bring that back and talk about how we look at rare disease drug development and how sometimes we're looking at things a little upside down and maybe not what we need to do with starting with establishing a good foundation. So here we have what we would think about how we should be viewing a house and how a house is built from the bottom up. And with that, we have traditionally, when we have drug development in our more common diseases, we are able to lay out a good foundation. We have an understanding of the disease course. We may be able to have preclinical models and animal models to help us understand aspects of the molecule we're studying. We may have a core set of symptoms or cardinal symptoms that we can focus in on and target. We often have large patient populations. We may have established endpoints or ways to measure these endpoints in the trials. But when we think about our challenges in rare disease drug development, we often have holes in our foundations. We don't have all of this needed information to really help us be able to optimize our clinical trials in our rare diseases. So often our natural history is often poorly understood, where we have unknown causes of disease. We have variation, heterogeneity of both the genetic and the phenotype of the disease, and where our patients may be exhibiting various symptoms, and are we actually able to select an appropriate endpoint or know what we should focus the study. Our diseases in the rare disease space are often serious and life-threatening and progressive. We often have a lack of established efficacy endpoints, and so trying to choose one that we think will work can be a challenge for us. And often, as Teresa brought up, we have small and dispersed patient populations. So being able to study every possible patient may be hard and challenging if patients are unable to get to clinical trial sites. So how do we really address these challenges? We need to do this is we need to start by building a solid foundation, by integrating data from multiple data sources into a central repository, and by building a platform that can enhance our clinical trial readiness in the pre-competitive space. And with this, we can ultimately foster innovation in our rare disease drug development arena. And that is what we really see RDCA-DAP being able to provide for us is this foundation? So this slide that you see is really what I call our hallmark slide of conceptually what we're trying to do. Our colleagues at the Critical Path Institute in NORD have put this slide together to really envision what we hope RDCA-DAP will accomplish. And today you're going to be hearing highlights of how this works and how all of our stakeholders are part of this and can help us by bringing different various sources of data. Why I like this slide is it really does show why we need everyone to contribute to this initiative, as well as how all of our stakeholders are critical through the entire process. Because if we're able to bring in various data sources, as you see on the left-hand side, we can ultimately be able to have actionable solutions that will impact us all. So what do we hope long-term will be the impact of RDCA-DAP on rare disease drug development? We hope that we'll be able to develop more efficient and effective clinical trial protocols. We hope we can encourage the standardization of data that can be extracted into our CDISC formats for regulatory submissions. And these are things that you'll be hearing about shortly in presentations from my colleagues. By pooling data together will allow us to have a better understanding of how a disease progresses across the range of patients. Can it help us optimize clinical trial protocols, which can include endpoints, inclusion criteria, and the size and power of the trial that we need? We hope from RDCA-DAP that we'll be able to have a set of analytic tools and simulation tools to help optimize a trial protocol. This may help us be able to help determine what is a sufficient sample size, for example. We want to have the ability to look at dynamics of change in our outcome assessments and our biomarkers in individual disease states, and help us understand the variation from the heterogeneity in most of these diseases and how that impacts the course of the disease and the rate of change that a molecule may have. And is it even possible that we may have the ability to potentially find and match historical controls to help enrich maybe a placebo arm and reduce the number of patients? These are all the goals that we hope that RDCA-DAP can accomplish. Up next, we'll be having a variety of presentations really showing the aspects of RDCA-DAP and what can be done. These presentations really show how working together with various stakeholders is important and critical with this initiative. And when we work together, we are able to advance the science and advance rare disease drug development.